Okay, growth and development, botany, chapter 11. Okay, so growth is defined as an irreversible increase in volume due to the division and enlargement of cells. And as the cell mature, they differentiate into forms adapted to specific functions. So development is a change in form as a result of both growth and differentiation. And cells themselves assume different shapes and forms, which allows volume to increase at a greater rate than the usual surface area. So many growth phenomena are influenced by hormones, which are produced in minute, which is really tiny amounts in one part of an organism and usually transported to another part where they have specific effects on growth, flowering, and other plant activities. So vitamins are organic molecules that function in activating enzymes, and they are sometimes difficult to distinguish from hormones. So this picture just shows you some examples of plant hormones and growth, and the control of the cell on the left, cell division, that is the cytokinins, and the gibberellins. Then you see the stomatal closure is due to the abscisic acid, and the development of the abscissa zone is ethylene and auxins. And then on the right, the control of the stem elongation is auxin and gibberellic acid, and the initiation of flowering or the flowering hormone from the leaves, and then the growth of young fruits, cytokinins, and induction of fruit ripening is ethylene. So Darwin and his son noted in 1881 that coleopters bend towards a light source. So Fritz Wendt in 1926 followed up on the Darwinian observations and demonstrated that something in coleoptile tips moved out into agar when decapitated tips were placed on it. And he called the substance auxin and showed that it can cause coleoptiles to bend. So this is just an example, and it shows the shaded side in the little cells up there. It shows the shaded side of the coleoptile and the illuminated side of the coleoptile. So you can see which side the light is coming to, and the fact that it will bend towards the light, but the cells are actually growing more on the other side. Okay, there are three types or three groups of plant hormones that promote the growth of plants that have been found. Others are known only for their inhibitory effects. So auxins stimulate the enlargement of cells and are involved in many other growth phenomena. And synthetic auxin-like compounds have been used as weed killers and defoliants. And dioxin, which has been shown to cause disease and defects in laboratory animals, is a toxic contaminant of 2,4,5-T, which is now banned in the United States. And you've probably heard of 2,4-D before too. Okay, so gibberellins promote stem growth without corresponding root growth. So stem growth without root growth. Cytokinins promote cell division, and they can be used to stimulate the growth of axillary buds. Abscisic acid causes buds to become dormant and apparently help the leaves respond to excessive loss of water. And ethylene gas hastens ripening of fruits and is used commercially to ripen green fruits. Senescence. This is the breakdown of cell parts that leads to the death of the cell, senescence. Okay. Plant movements such as nutations, not mutations, but nutations with an N-U-T-A-T-I-O-N-S, and nodding, twinning, contractile, and nastic moments are growth movements that result primarily from internal stimuli. This short little clip is an example of a nutation. See how it's growing here? So tropisms are permanent. They're directed growth movements that result from external stimuli such as light. So light is phototropism, gravity, which is gravitotropism, contact, which is thigmotropism, and chemicals, which is chemotropism. Turgor movements result from changes in internal water pressure. They may be very rapid or they may take up to 45 minutes to become visible. So turgor movements include contact movements and sleep movements in which leaves or flowers fold daily in what is known as a circadian rhythm, or other types of plant movements which include water conservation movements, taxes, and dehydration movements. This illustration just simply shows a plant when the turgor cell from a leaf has enough water and the plant is standing straight up, where it shows a flaccid plant where the cell from the leaf doesn't have enough water in it. So we say the plant is flaccid. Okay, photoperiodism is a response of plants to the duration of night. So short day plants will not flower unless the photoperiod is shorter than a critical day length. And a long day plant will not flower unless the photoperiod is longer. And then there are intermediate day plants that have two critical photoperiods. And then the flowering of day neutral plants is independent of the photoperiod. And phytochromes. These are light-sensitive pigments that occur in all higher plants, and they play a role in many different plant responses. So your phytochromes occur in two forms, each of which can be converted to the other by the absorption of light. So daylight generally results in more PR being converted to the PFR, which is the active form, than vice versa. So PR absorbs the red light and PFR absorbs the far red light. So PFR will convert back to P over a period of several hours in the dark. So PR is stable indefinitely in the dark. So the precise mechanism of photoperiodism is not fully understood, but phytochrome interconversions play a role in the photoperiodism, but it cannot be used alone to explain it. So in response to the photoperiod, the flowering locust gene produces a protein that is transported from the leaves to the shoot of the apical meristems. This induces flower production. So cytochromes have functionalities that are very similar to phytochromes. And each species of a plant has their own optimal temperature for growth. Okay, so we know that dormancy is a period of growth inactivity in seeds, buds, bulbs, and other plant organs, even when the appropriate environmental conditions are met. We know that some of those things will lay dormant. Quiescence is a state in which a seed is unable to germinate unless appropriate environmental conditions exist. So on the left, this germination requires fire, and this is of the lodgepole pine. And the cones open in the heat of fires, and the seeds require bare mineral soils to germinate. So therefore, that's why they need to open in fire to get that bare soil to germinate. And then on the right is an example. Let's see, it's Banksia, Hakea, and some eucalyptus plants bear their fruits with hard woody cases that are not dropped from the plant. And then the heat of a fire stimulates the fruits to actually open, and then the seeds are released. So the change from dormancy to a state in which germination will occur in seeds is controlled by a variety of different factors. These factors include temperature, moisture, photoperiod, thickness of the seed coat, enzymes, and growth inhibitors. So stratification is the breaking down of dormancy in seeds by keeping them refrigerated and moist for several weeks. So these are the type of seeds you see in the picture that we are used to seeing. I see corn, rice, flour, several other different seeds there. Okay, and once again, this is the reference for our text, and we will go over more review questions later. Thank you. See y'all later.